Welcome to Unburied Remnants. The official narrative is just the surface. We dig into what's underneath. And we're just getting started. "15% of the human population carries a blood type found in no other primate on Earth. Not in chimpanzees, not in gorillas, not in any of our closest genetic relatives. It is not a disease. It is not a deficiency. By every standard medical measure, the people who carry it are perfectly healthy. And yet, it behaves differently. It interacts differently. And in one specific biological context, it is incompatible with the other 85% in a way that natural selection should have eliminated thousands of years ago. Science knows what it does. What science cannot agree on is where it came from. This is the Rh-negative blood question, and it is one of the strangest unsolved puzzles in human genetics. To understand the question, you have to understand the biology. And the biology is genuinely strange. Your blood type is determined by antigens, proteins on the surface of your red blood cells. The ABO system you already know, type A, B, ABO. The Rh factor is a separate system, a different protein, discovered in 1940 when researchers were working with rhesus monkeys. Hence the name, rhesus factor, Rh. If you have this protein on your red blood cells, you are Rh positive. If you do not have it, you are Rh negative. Simple enough. Except for what follows. When an Rh negative person is exposed to Rh positive blood through transfusion or through pregnancy, their immune system does something remarkable. It recognizes the Rh protein as foreign, as something that does not belong, and it builds antibodies against it. The first exposure is the sensitization. The immune system learns. The second exposure is the response, and the response can be severe. This is where the evolutionary puzzle enters. Every trait that reduces reproductive success should disappear within a few generations. Natural selection is ruthless about this. A blood type that creates incompatibility between a mother and her own child, that causes the immune system to treat the next pregnancy as a threat, should not survive. The mathematics of natural selection are unforgiving. And yet here it is, in 15% of the human population, still here. After thousands of years of recorded human history, and almost certainly long before that, there are three possible explanations mainstream science offers. A random mutation that somehow persisted despite its reproductive cost. An ancient isolated population that carried it and merged with the broader human family. Or a genetic lineage that did not originate in the same place as the rest of human DNA. The third explanation is the one mainstream science is least comfortable with, because the distribution of Rh-negative blood across the world is not random. And once you see the pattern, it is very difficult to explain away. If Rh-negative blood were a simple random mutation, you would expect to find it distributed more or less evenly across human populations worldwide. Random mutations do not respect geography. They do not cluster. But Rh-negative blood does cluster, significantly. The highest concentration in the world is found among the Basque people of northern Spain and southwestern France. Between 30 and 35% of Basques are Rh-negative, more than double the global average. This is not a small statistical variation. It is a striking outlier. The Basques are themselves one of the great mysteries of European prehistory. They speak a language, Euskara, that is unrelated to any other language on Earth, not Indo-European, not Semitic, not related to any language family that has ever been identified. It is a linguistic isolate, a language that appeared or survived from somewhere we cannot trace. Beyond the Basques, Rh-negative concentrates in specific populations across the ancient world. Certain populations in the British Isles, the Atlas Mountains of North Africa, specific groups in the Middle East and the Caucasus region. It does not distribute evenly across Asia or sub-Saharan Africa, where rates drop to 2 or 3% or lower. The distribution looks less like a random mutation spreading outward from a single point and more like the signature of an ancient population that once occupied a specific region and whose descendants are still carrying the marker. Which population? Which region? How long ago? These are the questions the distribution data raises and does not answer. Stay with me here, because the three explanations science offers for Rh-negative blood are very different in their implications, and only one of them accounts for everything we have just looked at. The first explanation, random mutation. At some point in human prehistory, a random genetic mutation produced the Rh-negative phenotype. Through genetic drift, the random fluctuation of gene frequencies in small populations, it survived and spread. This is the simplest explanation. It requires no unusual assumptions. The problem? The distribution. Random genetic drift does not produce the pattern we see. Extreme concentration in one specific population, moderate concentration in adjacent populations, near absence everywhere else. Drift produces smoother distributions. What we have is a spike. Spikes in genetic data suggest founder effects, a small founding population that carried the trait at high frequency. The second explanation, ancient isolated population. A population living in geographic isolation, perhaps in the region of the Basque Country or the broader Atlantic coast, carried Rh-negative blood at high frequency. When they interacted with or were absorbed by the expanding Indo-European populations of prehistoric Europe, they contributed their genetics to the broader gene pool. Their Rh-negative marker persisted. The Basques, who resisted linguistic assimilation, preserved the highest concentration. This explanation is more sophisticated and more consistent with the distribution data. It has serious genetic support. The Basques do show other genetic markers that distinguish them from surrounding European populations, not just the Rh factor. There was a real ancient population here that was different. What it does not explain, where that population came from, what produced the Rh-negative marker in them in the first place, why their language is unrelated to any other on Earth. The isolated population theory moves the question back one step. It does not answer it. The third explanation is not mainstream, but it is not fringe either. It follows directly from the data, taken seriously. If Rh-negative blood does not appear in any other primate, not in the species we share 98% of our DNA with, then where did it enter the human genome? The standard answer is mutation. But if the mutation explanation has distributional problems, and the isolated population explanation only pushes the question back, what is left? What is left is the possibility that Rh-negative blood entered the human genome from a source that was not part of the standard human genetic lineage, not a mutation within the species, an introduction from outside it. Mainstream genetics does not go here. The tools of population genetics work within known lineages. They do not have a framework for evaluating the possibility of genetic introduction from an unknown source. So the question simply is not asked in those terms. But there is a tradition that does ask it, and it has been asking it for a very long time. This is the part I want to be careful about, because what follows is not science. It is pattern recognition between what the genetic record shows and what the oldest texts in human history describe. And the overlap is uncomfortable. In episode 2 of this series, we examined the Book of Enoch. The Watchers, divine beings who descended, took human partners, and produced offspring that were described as different. Hybrid. Neither fully one thing nor fully another. The Nephilim tradition across cultures is consistent on one point above all others. The offspring of these unions were physically distinct, described as giants in some traditions, described as having unusual features, different eyes, different hair, different skin, different in ways that persisted across generations. A visible marker of mixed origin, the Sumerian tradition describes the Anunnaki interacting with humanity directly. The result in some texts is a modified human lineage, not fully human, not fully divine, carrying something forward that was introduced rather than evolved. The Hebrew tradition uses the phrase sons of God, Beni ha-Elohim, for the beings who produced the Nephilim. In the divine council framework we examined in episode 3, Elohim refers to beings of the divine council, not human. The offspring of that union carried something that marked them as different. Now, the genetic question. If the Watcher tradition is describing something that actually happened, beings of non-human origin interacting with the human population and producing offspring, what would we expect to find in the genetic record? We would expect a genetic marker that does not appear in other primates. We would expect it to concentrate in specific ancient populations rather than distributing evenly. We would expect it to persist despite reproductive pressure against it. We would expect the populations that carry it at highest frequency to show other signs of unusual origin, like a language completely unrelated to any other on Earth. I am not telling you that Rh-negative blood is the genetic signature of the Nephilim. That is a claim the evidence cannot support. What I am telling you is that the pattern the Rh-negative distribution produces, in the genetic record, in the population data, in the linguistic evidence, is consistent with the introduction of a genetic marker from a source outside the standard human lineage, and that the oldest texts in human history describe exactly that kind of introduction, at exactly the time we would expect, in exactly the regions where the concentration is highest. 3 data points, 3 independent sources, the same pattern. Draw your own conclusions. I want to be upfront with you about what we know and what we don't, because the responsible version of this question matters as much as the interesting version. Here is what the science actually establishes. Rh-negative blood is a real genetic variant. Its distribution is genuinely unusual and not fully explained by simple random mutation. The Basque population does carry it at significantly higher rates than any other population on Earth. The Basque language is genuinely unrelated to any other known language. These are facts. Here is what the science does not establish. There is no peer-reviewed genetic study that supports the hypothesis of non-human genetic introduction. The tools of population genetics work within known lineages, and the Rh-negative distribution, while unusual, has not been formally classified as unexplainable within standard evolutionary frameworks. Unusual is not the same as impossible to explain. Here is where we are left. A genetic marker that does not appear in other primates. A distribution that clusters in ancient populations in ways that look more like a founder effect than a random mutation. A linguistic isolate at the center of that distribution. And a set of ancient texts that describe, in considerable detail, the introduction of non-human genetics into the human population, in the same region at approximately the same time. None of this is proof. All of it is pattern, and the pattern is consistent. If you are Rh-negative, you are carrying something that has been here for a very long time. Something that persisted when it should not have. Something that the oldest stories in human history may have been trying to describe. What that something is, science has not yet fully answered. Across 10 episodes, we have followed a thread. It began in the clay tablets of Sumer, with gods who came before, and a flood that reset everything. It moved through the physical archaeology, Göbekli Tepe, the underwater sites, the king list. It arrived at the genetic record, a marker in 15% of the human population that nobody can fully explain. Every discipline points in the same direction: the textual evidence, the archaeological evidence, the geological evidence, the genetic evidence. None of it proves what happened before the flood. All of it is consistent with a world more complicated than the official narrative allows. 2 episodes remain in Season 1. Episode 11 looks at what didn't make it into the Bible, and why. Episode 12 asks the question the whole season has been building toward. If every culture on Earth tells the same story, what does that mean? Next episode leaves the genetics entirely. Because there is one more layer of evidence we have not examined, not in the ground, not in the blood, in the texts themselves. Specifically, the texts that were removed, excluded, decided against by specific people in specific rooms for specific reasons. Episode 11. The Missing Texts, What Didn't Make the Bible and Why. Subscribe so you don't miss it. And if this episode made you think, share it. The algorithm rewards engagement in the first 24 hours. Every view, every comment, every share makes a difference for a channel this size. Thanks for watching Unburied Remnants. If you like the content, please like, comment, and subscribe to support the channel. Stay curious. Keep digging. The rabbit hole goes deeper.